Hi, my name is Jack Foley. I'm a senior biology student at Framingham State University, and I worked with Dr. Avard on developing an analysis of DESI mass spectrometry imaging for the invasive fronts of tumor spheroids. So why study breast cancer? Well, it's the most common cancer among women, the second most deadly cancer in women, and metastasis, or the ability of cancer cells to migrate, is the leading cause of death in cancers, not just breast cancer, but all cancer, and up to 90% of cancer deaths are from metastasis and not the original tumor itself. Lipids play an important role in the migration of those cancerous cells, from energy to the cellular membrane, cellular signaling, and hormones, but there are thousands of lipids in a cell and thousands of lipids in a cancerous cell. How do we analyze them efficiently? That's where mass spec comes in. This works through spraying a solvent at a sample forcing ionization that's detected and categorized by the mass to charge ratio, or M/Z. One of the major advantages that mass spec has over other methods is it's a high throughput and very accurate in distinguishing different molecules within one sample. So when we do mass spec, what are we looking at? We're going to be looking at tumor spheroids grown in 3D matrices. Compared to 2D, that better represents the human body environment. And this is what a confocal image of a tumor spheroid looks like. This white represents the core, this red circle represents the invasive front, or those cells starting to migrate away. This is a sample of what one of the images looks like in mass spectrometry. The red meaning there's a higher concentration, and the purple meaning there's a lower concentration. And this white circle represents the tumor spheroid core, and this red represents the invasive front. But previous work has really only focused on the cores. Why does the invasive front matter? Well, like I mentioned before, the invasive front are those cells starting to migrate away. And if we're looking at this core image, the core really has its most intense signal at a high concentration. The invasive front has no signal in the core, it's just the invasive front at a lower concentration. And this image shows the core with an invasive front intensities in both locations. The really cool thing about these images are that they come from the same spheroid. The only difference in what we're looking at is a different molecule. So the image on the left of the core, that molecule is only expressed in the cells of the core. In the middle of the invasive front, whatever molecule that is only being expressed in the cells in the invasive front migrating away. In the core and invasive front, whatever molecule that is being expressed in both locations. And all these images came from HDI, or the image processing for mass spectrometry. So how do we differentiate core from invasive front? The first thing we do is we find a core image in HDI. It'll look something like this. We convert to grayscale because the computer does not really know what to do with all the color. And then we wrote a code in JavaScript so that it automatically will crop the image. It'll automatically threshold it so all the pixels are on or off. And then we can then automatically find the center and place a pinwheel in the center of that object and begin running an analysis on each line starting in the middle going out. In this graph on the right, it shows that sudden drop off from where the core, or the white, suddenly meets the black, or everything after the invasive front, giving us a predetermined core that we can use for every image in the same spheroid following. So say we want to use this image for an analysis, we bring it from HDI, we convert it to grayscale, and then our second code in JavaScript, it will pull those coordinates from before and place that same pinwheel on it and begin running that same analysis starting in the center of each line and going towards the out, or going out from the cell, giving the intensity for each micron along that line. And this yellow line shows us that core sudden drop off, so everything to the left is going to be defined as the core, and everything to the right is defined as the invasive front. So is there really a difference between the two? Both of these graphs come from the image in the top right. Each of these lines are the same 16 lines in the same image. They're just broken up to show a little clarity. The green on the left is the core, and The orange on the right is the invasive front. As you can see on the core, there's a sudden drop-off in intensity and then nothing afterwards. And since each drop-off is at a slightly different point, it's why it's important that we have that predefined core, so we don't have to worry about eyeballing it, we don't have to worry about making mistakes. And then the invasive front is everything after that first drop-off. And it's relatively consistent no matter how far outward from the core it is. So how do we know that our code actually works? We can take different samples of images and look at them separately. So for this core image, there's no invasive front. The signal's relatively strong, so when we look at a graph, we see that the signal's only coming from the core, and there's no signal in the invasive front because there is no invasive front. If we look at the invasive, we expect to see almost the opposite. There's not really any core that we can see, but since we have that predefined core, there's a small amount of signal, but the majority of the intents and signals that we're seeing come from the invasive front. And if we look at an image of a core and invasive front, we expect a mixture of the two graphs, and there's a relatively equal distribution of signal, however, but not intensity, but it still shows that both the core and invasive front are being picked up and able to be distinguished. So now that we know the code works, what do? Well, we can use the code we've written in the methods described throughout and apply it to other samples. For example, we've begun looking at the impact mutant P53 has on lipid expression and distribution in cancer. We expect some pretty interesting results because very early analysis shows a mutation of P53 can impact that lipid distribution, including oleic acid, which is shown here in cells with a mutant P53. I just want to thank you all for taking the time to watch this. I also want to say thank you to my advisor, Dr. Avard, the Kaufman Group at Columbia University, and the Framingham State University Biology Department.